Thank you very much, Alice, for that very nice introduction. And I'd like to thank the Society for the honor of inviting me to give this lecture. So I'd like to tell you about a tale, really, from our lab about the roles of Wnt signaling in development, regeneration, and disease. And I have no conflicts of interest to disclose. So we feel that understanding the key events in skin development and renewal is really important for really understanding how these events go awry in disease, and in particular for identifying new therapeutic approaches to both common and rare skin diseases. So the epidermis of the skin starts off life as a single-layered surface ectoderm, shown here in pink. And this gradually stratifies during embryonic development to form the epidermis, which obviously provides a protective barrier for our body. In addition, the epithelial and mesenchymal cells of the embryonic skin interact to form a wide variety of different ectodermal appendages. And the first step in appendage development is generally a localized thickening in the epithelium known as a placode. And then these then go on to develop into mature organs such as hair follicles. In adult life, the skin epithelia are renewed due to the activity of stem cells. And defects in these processes lead to diseases such as skin cancers and common conditions such as male pattern baldness, as well as genetic dysplasias. So studying the embryonic events involved in appendage development in particular are really relevant to understanding the adult renewal of these organs, because we find that similar molecular and cellular events underlie both development and renewal. And understanding these processes also has practical applications. So for instance, we currently treat severe burns and wounds in the skin by grafting of keratinocytes, and this is very effective in regenerating the epidermis, but the skin grafts lack appendages such as hair follicles and sweat glands, and are so not fully functioning. By understanding the embryonic events that induce the formation of these structures, we expect to be able to inform better approaches to regenerating fully functional skin. So for these reasons, one of the questions we've been interested in my lab for a long time is what the signals are that initiate the development of ectodermal appendages from the surface ectoderm, rather than just having the ectodermal cells simply stratify to form an epidermis. And a clue to one of these really important pathways came when we and others found that transgenes that report activity of the Wnt beta-catenin signaling pathway reveal really early activity of this pathway that's localized as appendages emerge from the skin. And here I'm showing you a picture of hair follicle development in mouse embryos, and you can see I guess this doesn't work too well, but you can see the hair follicle placodes, these spots appearing in the ectoderm that are dark blue, indicating high levels of Wnt signaling. And similarly, tooth placodes also show high levels of Wnt activity, and we also see Wnt activity locating to fungiform taste papilla placodes in the dorsal tongue. So Wnt's are secreted ligands that activate the beta-catenin pathway by interacting with frizzled receptors and LRP co-receptors, and this serves to inactivate a complex of proteins that normally phosphorylates beta-catenin in the cytoplasm and targets it for degradation. In the presence of a Wnt signal, the beta-catenin accumulates in the cytoplasm and moves into the nucleus, where it forms complexes with transcription factors of the left TCF family. These activate transcription. And later on in my talk, I'm going to discuss how these complexes interact with other transcription factors in order to achieve specificity in the outcome of Wnt signaling. This pathway can be inhibited by secreted proteins such as DKK or DICOP family members, and DKKs work by interacting with the LRP Wnt co-receptor and causing its internalization. So in order to determine whether this pathway is functionally relevant in different aspects of skin development, what we've done is to genetically manipulate different elements of this pathway. So for instance, when we deleted beta-catenin in the epithelial cells of mouse embryos, we found that this completely blocked the initiation of hair follicle development. And similarly, if we force expression of the DKK inhibitor, DKK1, this blocks almost all fungiform taste papilla formation and also prevents the development of mammary glands. Conversely, if we mutate beta-catenin into a constitutively active form that can't be degraded, this results in the formation of multiple ectopic appendages such as taste papillae and teeth, and also causes the surface ectoderm to inappropriately produce hair shaft keratins instead of stratifying normally. So these experiments showed us that Wnt beta-catenin signaling is really a fate-determining factor for the surface ectoderm that promotes the formation of appendages. So as we all know, the distribution of hair follicles varies a lot by body region. And most dramatically, on the skin of our palms and the soles of our feet, there's a complete lack of hair. This is also true in the mouse, which has a hairless plantar region, and even in animals like polar bear that develop quite a bit of hair on the soles of their feet, they also have hairless regions. So we've been very interested in what the factors are that determine the formation of hairless versus hairy skin. And a clue to this problem came when we found that another member of the DKK Wnt inhibitor family, DKK2, is expressed in hairless plantar but not hairy dorsal skin in mice at late stages of embryogenesis and around the time of birth. And in the higher magnification picture, you can see that DKK2 is expressed at high levels in the dermis of plantar skin. And we confirmed this also at the RNA level using quantitative PCR that showed much higher levels of DKK2 mRNA in plantar versus dorsal skin. So to determine whether this expression of this Wnt inhibitor has functional consequences, we analyzed mice that lack DKK2, and we first crossed these two axin 2 lac C mice that express beta-galactosidase at sites of Wnt signaling. And what we found is that in wild-type mice, the plantar dermis has low levels of Wnt signaling, but in DKK2 null mice, we see this activation of the pathway. And this leads to ectopic formation of hair follicle placodes that express LEF1. And ultimately, these placodes give rise to fully functional hair, as we can see in this light and scanning EM pictures of the plantar region of a DKK2 null mouse compared to a heterozygous control. So to summarize what I've told you so far, Wnt beta-catenin signaling initiates appendage development in the skin and in the oral cavity. The endogenous Wnt inhibitor DKK2 blocks hair follicle development in a plantar skin, and this gives us the first insight into how hairless versus hairy regions of skin develop during embryogenesis. We also think that controlled activation of this pathway may be useful in combination with tissue engineering approaches for regenerating hair follicles and teeth for therapeutic purposes, and this is something that we're currently exploring in the lab. So during adult life, epithelial organs are renewed. As we all know, hair follicles go through cycles of rest, growth, regression, rest, and regrowth. And most of our other epithelia, such as the epidermis and the sweat gland ducts, turn over continuously. Epithelia in the oral cavity also turn over, including the filiform papillae that give our tongue its rough surface, and taste buds which lie within taste papillae, such as the fungiform papillae. So it's thought that this ability of epithelia to regenerate relies on stem cells that can both self-renew and give rise to differentiating progeny. And we're really interested in understanding the signals that allow these processes to happen. So a clue to this issue came from actually genetic analysis of human patients, which showed that the human Wnt10A gene, when mutated, is associated with late onset of ectodermal defects, suggesting that Wnt10A might be an important Wnt ligand for epithelial renewal. So in these patients with severe loss of function mutations in Wnt10A, we see adult onset of defects such as thinning hair, palmoplantar keratoderma, and a smooth appearance of the tongue, as well as a partial or sometimes even complete lack of secondary dentition. In addition, a variant of Wnt10A that's associated with lower expression levels has been associated also in GWAS studies with male pattern baldness. And Wnt10A mutations are actually surprisingly common, occurring in 1 to 3% of the population, and over 50 disease-associated variants have been described. That we were very fortunate to be able to collaborate with Emily Chu in our department, and Emily has a patient who's homozygous for a predicted loss of function mutation in Wnt10A. And this patient exhibits severe hypodontia, palmoplantar keratoderma, and nail defects. And we tested this lady's ability to sweat by applying a starch iodine mixture to her palms, and in controls, this results in the deposition of these brown grains that as a result of reaction with the sweat. And in this patient, though, was completely unable to sweat on her palms, even when we got her really hot by putting her up against a radiator. So the mutation in this patient is predicted to prevent a splicing of intron 3, and we confirmed that this is in fact the case by PCRing the RNA from her plucked hair follicles, and we found that indeed there was no product of splicing of intron 3. And this results in a predicted production of a protein that lacks most of the conserved residues that are required in Wnt proteins for frizzled receptor binding. So we think it's likely a loss of function mutation. So consistent with the defects seen in the human patients, we find, now turning to mouse tissues, that Wnt10A is expressed in the epithelia that are defective, and here we can see that Wnt10A is not expressed in resting hair follicles, but comes on at anagen onset and is highly expressed during the growth phase, as well as being highly expressed in the epidermis and sweat ducts, and in taste and filiform papillae in the tongue. So in order to understand the signaling properties and functions of Wnt10A in more detail, we decided to generate a loss of function mouse model. And we made a floxed allele of Wnt10A that we then crossed to a globally expressed Cree construct for global deletion of Wnt10A. We also crossed it with a keratin 14-driven Cree in order to delete Wnt10A in epithelial cells, and to a K5-driven reverse TET transactivator paired with a TET-O Cree construct. And what this allows us to do is to induce deletion of Wnt10A at any time that we want, specifically in epithelial cells, by treating the mice with doxycycline. And this is important because it allows us to distinguish possible functions of Wnt10A in regeneration from its functions in development. So we were happy to see that the mutant mice nicely recapitulated phenotypes seen in the human patients, including dental defects such as flattened cusps of the molar teeth, failure of bifurcation and extension of the roots of the teeth, and also low bone density in alveolar bone that was seen in both the humans and the mouse. So these defects arose during embryogenesis. But interestingly, and consistent with the adult onset of other defects, we found that the development of other appendages was not affected during development by loss of Wnt10A. So hair follicle, taste, sweat glands, and tongue fully formed papillae were all relatively normal at birth. However, during adult life, we started to see dramatic defects, including loss of hair, similar to the thinning of hair in the patients. This smooth tongue was also recapitulated in the Wnt10A mice, and they showed defects in nail growth. So mice, unlike humans, only develop sweat glands in a very restricted region on their foot pads. So to analyze sweat gland formation and function, we used foot pad skin, which we separated with disphase 2 and then stained with Nile blue. And here you can see that deletion of Wnt10A in adult life leads to dystrophic sweat ducts, which kind of shrink up and fail to maintain their normal length. And this results in functional inability to sweat, which we tested with starch iodine, just as in the human patients. And we similarly found that forced expression of the DKK1 Wnt beta-catenin inhibitor also prevents sweating, suggesting that Wnt10A might be signaling through the beta-catenin pathway. To test this directly, we crossed the Wnt10A mutant mice, 2 mice expressing a Wnt reporter gene that results in GFP expression. And you can see that unlike in control hair follicles, Wnt signaling is much reduced in mutant hair follicles and also in taste buds and other organs that are defective in the mice and the human patients. So the defects that we see seem to be due in part to decreased proliferation. So the mutant mice displayed decreased proliferation that was statistically significant in all of the epithelia that we examined. So these included hair follicles which showed delayed anagen onset and markedly reduced proliferation. And with age, these hair defects became increasingly severe, ultimately leading to a miniaturization of the epithelial portion of the follicle and enlargement of the associated sebaceous glands. And this was interesting because of the GWAS studies that implicates Wnt10A in male pattern baldness, because the appearance of bald scalp is very similar to what we're seeing in the mice. We also see that hair follicle stem cell markers are retained even in this degraded epithelium. And this is also similar to what Luis Garza and George Kotsarellis have found in bald human scalp, which similarly retains hair follicle stem cells. So the proliferative defects in these mutants led us to ask whether Wnt beta-catenin activity marks self-renewing stem cells in these different epithelia. To address this question, we used mice in which an inducible form of Cre recombinase has been knocked into the axin 2 locus, and axin 2 is a ubiquitous direct Wnt target gene. We paired this construct with Cre reporter genes that express fluorescent proteins when Cre is activated. So the strategy here is to induce Cre activity for 1 or 2 days using tamoxifen, and this will then label any Wnt-active cells with fluorescent markers, and then we can come back and look at this tissue after many cycles of renewal to determine whether it's still labeled. So if stem cells are initially labeled by Wnt signaling, then we would expect after many cycles of tissue renewal that these tissue would remain labeled and that we would see continuous labeling of the stem cells and their progeny. Whereas if Wnt activity is confined to differentiating cells, we would expect this label to be lost. So this is an example of one of the experiments in which we examined Wnt activity in tongue and plantar epithelia, and you can see that there are these nice, stably maintained clones of labeled cells, indicating that there are indeed Wnt-active self-renewing stem cells in the basal layers of these tissues. And similarly, in the nail, we were able to see these beautiful clones of labeled cells emanating from the nail matrix and giving rise to the nail plate and the nail bed. So these data that I've shown you indicate that Wnt10A is a really broadly acting ligand that drives progenitor cell proliferation in multiple adult epithelia. However, a general decrease in proliferation cannot entirely explain some of the really striking phenotypes that we see in Wnt10A patients. And these include the palmoplantar keratoderma, which, as you can see, is very regionally restricted to the palms and soles of the feet, whereas the wrist skin is entirely normal. So in trying to think of what are the factors that are really different between palmoplantar and other skin, we were drawn to the keratin 9, which is specifically expressed in the palms and soles, and when mutated, gives rise to severe epidermal defects. And indeed, we found when we examined keratin 9 expression in human patients with two different Wnt10A mutations, that keratin 9 protein was dramatically reduced in the patient plantar skin, shown here, compared with the control. Whereas another broadly expressed keratin 10, was not affected. And using a biopsy from our human patient, we could show that our patient at Penn, we could show that this reduction was at the mRNA level. And similarly, in mice lacking Wnt10A or induced for loss of beta-catenin, there's a big decrease in keratin 9 expression in foot pad epithelium, which is reflected at the mRNA level. We also see defects in differentiation in the tongue filiform papillae, which normally express these hard keratins, and these are absent in the mutant despite the fact that rudimentary papilla structures are present. And what we found is that HOXC13, which is a transcription factor that is necessary for filiform papilla differentiation, is absent if we knock out either Wnt10A or beta-catenin. And we also see that LEF1, which marks the proliferating cells in these structures, is also dependent on Wnt signaling. So this led us to the question of how does Wnt signaling regulate these two apparently opposite functions of proliferation and differentiation. And our hypothesis is that beta-catenin complexes with different transcriptional partners in proliferating versus differentiating cells, and that these partners are not dependent on Wnt signaling, but are there already regulated by other factors. So a strong candidate for such a partner is the transcription factor KLF4, which is known to be important for epithelial differentiation, and its expression is largely confined to suprabasal cells, for instance here in the foot pad epidermis, and it's not expressed in the basal layer. And it indeed overlaps with nuclear beta-catenin in these suprabasal cells. And when we delete beta-catenin, KLF4 expression is unaffected. So importantly, when we deleted KLF4 in adult life, we found that it had very similar effects to loss of Wnt10A, including the palmoplantar defects and this smooth tongue. And this similarity was also borne out at the molecular level, where we found dramatically reduced keratin 9 expression in foot pad epidermis lacking KLF4, whereas keratin 10 was not perturbed. And again, this decrease in keratin 9 was at the mRNA level. We also found that LEF1 is not expressed at all in foot pad epidermis, unlike in the tongue and in dorsal skin, for instance. However, another LEF TCF family member, TCF3, is expressed in both basal and differentiating suprabasal cells and overlaps in the suprabasal cells with KLF4. So we then wanted to know whether beta-catenin actually interacts directly with KLF4 and TCFs in differentiating cells. And for this, we used a proximity ligation assay, and this employs antibodies to the two proteins that are of interest. And these are tagged with different oligos that can be hybridized by a probe that contains complementary sequences for both. So if these proteins are within 40 nanometers of each other, this hybridization can occur, and then we then ligate the ends of the probe and can amplify it by rolling circle amplification, and this will give it a signal that appears as red grains. So here we're looking at the interactions of beta-catenin and KLF4, and you can see that there's a very nice signal in suprabasal but not basal footpad epidermis, indicating that these factors do indeed directly interact. And this interaction is absent if we delete beta-catenin, indicating that it's specific. And similarly, we find direct interactions of KLF4 with TCF3 in these suprabasal cells, whereas beta-catenin interacts with TCF3 in both basal and suprabasal layers. So we then wanted to know whether these complexes actually directly regulate keratin 9. And consistent with this idea, we found binding motifs that are conserved among species for KLF4 and for TCF left factors downstream of the keratin 9 promoter. And by CHIP qPCR, we could show that KLF4, TCF3, and beta-catenin all bind to this region. So this leads to a model in which in basal cells that lack KLF4, beta-catenin complexes with TCF3 to promote proliferation, whereas in KLF4 positive suprabasal cells, beta-catenin now complexes with KLF4 as well as TCF3, and then this can drive expression of differentiation genes that are specific to palmoplantar epithelium, such as keratin 9. And similarly, analyses of the filiform papillae showed that beta-catenin and LEF1 directly interact in these basal cells, whereas in the differentiating cells, we now find interactions of beta-catenin with another LEF family member, TCF4, as well as with KLF4, which we think drive differentiation. So to summarize the second part of my talk, I've shown you that beta-catenin signaling marks self-renewing stem cells in diverse adult epithelia, and we've identified Wnt10A as a very important ligand in driving epithelial proliferation. But we additionally find that beta-catenin is also critical for differentiation, and it performs this different function by complexing with cell type and region-specific transcription factors. And importantly, our analyses indicate that activation of the beta-catenin pathway in Wnt10A patients could be therapeutically beneficial. So just to give a little overview of what I've told you today, we've shown that during development, Wnt beta-catenin signaling is critical for appendage initiation, and this has potential applications in tissue engineering. We've also identified for critical functions for beta-catenin in promoting proliferation, and data from us and many other labs suggest that inhibiting this pathway could be useful in hyperproliferative conditions. And finally, we've identified Wnt10A beta-catenin signaling as being important in hair follicle growth, and our data suggest again, that stimulating this pathway could be useful in treating Wnt10A null patients, and possibly also in male pattern baldness. So I'd like to thank the members of my lab, in particular Minggang Zhu, who's here at this meeting and is a really talented postdoc fellow who's spearheaded the Wnt10A studies that I told you about. He was assisted by Jeremy Harrell, who is now a graduate student at Brown, and Emma Sung was involved in the DKK2 analyses. And I'd also like to thank all the other current and former members of my lab that have contributed to this work, as well as our many collaborators and our funding support from NIAMS, NIDCR, and the Dermatology Foundation. Thank you all for your attention.